Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today who is helping to create a better tomorrow for all of us on many different fronts. We have the honor of being joined today by Dr. Ashani Weeraratna, who is Bloomberg Distinguished Professor, Chair of the Biochemistry and Molecular Biology Department, Professor at Johns Hopkins Bloomberg School of Public Health. Dr. Weeraratna is also recently appointed member by President Joe Biden of the National Cancer Advisory Board, which advises and assists the director of the NCI on activities of the National Cancer Program. Dr. Weeraratna is a groundbreaking researcher exploring crucial questions about the role that aging plays in cancer, and her findings on age-related differences in responses to both targeted therapies and immunotherapies for cancer are really leading major change in clinical practice. She studies molecular mechanisms involved with the metastasis, ultimately how tumor microenvironments affect melanoma progression, as well as therapy resistance. She's very interested in how aging microenvironments guide and lead to increased metastasis and therapy resistance, as well as autonomous aspects of therapy resistance. And her research also encompasses biophysical changes that affect the ability of both tumors and immune cells to migrate. She's demonstrated the normal age-related changes in microenvironment can contribute substantially to multiple aspects of melanoma formation, then development, as well as therapy resistance. Her work in melanoma prevention actually includes public health approach as well, and she recently helped lead a campaign to introduce sunblock dispensers throughout Philadelphia here and ultimately talk to children about the dangers of tanning. In addition to all that, she is also an accomplished author with her book, Is Cancer Inevitable?, which describes not just cancer mechanisms, but how teams like hers are finding new ways to reduce the deadly impact. And this book is part of the Johns Hopkins Wavelength series. Dr. Weeraratna joined Johns Hopkins as a Bloomberg Distinguished Professor in 2019. She was originally at the Worcester Institute here in Philly, where she established her Microenvironment lab back in 2011. Prior to that, she was at the National Institute of Aging since 2003. Her doctorate is in molecular and cellular oncology from George Washington University Medical Center. She completed postgraduate training, was a postdoc fellow in experimental therapeutics and pharmacology at Johns Hopkins Sydney Kimmel Comprehensive Cancer Center, then known as Johns Hopkins Oncology Center. And we are lucky to have her with us today. Dr. Ashani Weeraratna, thank you so much for taking the time out of your schedule.
Thank you so much for having me and for that very kind and comprehensive introduction.
It's a fascinating story. It's a fascinating book as well. And one thing I didn't touch on in the intro is sort of the journey before your journey. And I have to say, from Sri Lanka to Zambia to Lesotho to Maryland to Philadelphia and back to Maryland, I don't think I've had anyone on a journey that's gone that far. Take us through some of those early days and when you first discovered along that journey that biochemistry and molecular biology was kind of cool and you wanted to learn more about it.
So I was born in Sri Lanka, and when I was 1, we moved to Zambia, which is in Central Africa. And then just a couple of years later, we moved to Lesotho. And Lesotho is this tiny little country landlocked by South Africa, for anyone who doesn't know. And I spent most of my formative years there. I left when I was 17 to come to America for college. And it was interesting because I grew up there during the time of apartheid, and in a way that really shaped my decision to come to the US, because when I was 16, I knew that I wanted to do cancer research, right? Which everyone is like, oh, that's kind of weird that you knew that already. But I loved biology. I loved, one of the first early experiments that I did as a teenager was to, they made us go outside and fish up some basically pond scum and put it in a petri dish and then culture it over a few days and see and identify some of the microorganisms that arose. And, we had to sketch them in a little book. And so I became very fascinated with bio in general. And then after that, I became very interested in molecular biology and did a lot of reading. And someone who was very close to me passed away of cancer. And, that's when I decided, well, I'd like to know more about this disease and try to understand it better. And so when I decided to go to college, my parents, who had grown up in sort of a very Anglophilic culture, said, okay, fine, you can go to the UK for university. And I said, no, I want to go to the US because that's where some of the most exciting discoveries are being made right now. I was 16. What did I know, honestly, at that point? I mean, there were probably just as many exciting discoveries in Europe, but in my head, that's where I wanted to go. And so I left and I came to St. Mary's College of Maryland, which is a tiny little college in Southern Maryland. And the reason that I ended up there was because my mom said, well, okay, fine, you can go to America, but you have to go somewhere near a family friend of theirs, my Auntie Cynthia, who became then a very close, they're basically my family here in the US. But, St. Mary's was a good 2.5 hours from DC, and it wasn't very easy to get back and forth. But, on holidays, I was always with them. So that's how I got here, and that's how I started at St. Mary's. And it was interesting because St. Mary's was sort of a very homogenous school at the time. I think it's changed a little bit since then, but everybody was from suburban Maryland, basically, except for me. So I was their token international student. And it was interesting because everyone was so welcoming and so kind. And in a way, it was like shock therapy for me. I was just like immersed into American culture immediately. And I got to learn a lot about American culture and Maryland and, the area in general.
Wonderful. Wonderful. Yeah, it's, as I said, looking at just those first couple of pages in the book, it was pretty fascinating to see the journey that you took before starting this journey. But it was fun reading about that too. I'd love to start off, if we could, because, there's so many different parts of your story that it weaves a very elegant tapestry, I have to say, taking up to exactly what you're doing now. But I'd love to start off sort of going back about 20 years when you were at the National Institutes on Aging, because it was here that you started to publish some of your work on melanoma and WNT, or Wnt signaling, which brings up a fascinating topic on its own because, we've discussed cancer on previous shows. We talk about, sort of these bad genes here, these oncogenes, these good ones over here, these tumor suppressors. But you're working with a set of genes that had a very sort of interesting double-edged sword component, implicated in carcinogenesis, but at the same time, we're learning all this stuff about how these important WNT genes, if we don't have them, we don't form in the first place. They're responsible for our body patterning, cell fate, cell migration. Talk a little bit about this, because I think we think about sort of this pleiotropy, or what we'll call it, that, not all these genes are good or bad. Some have these double-edged sword components or double functions, and we really have to take that into account when we think, and we'll be going the tumor microenvironment a little bit, when we think about cancer. Talk a little bit about that, if you would.
Yeah, I mean, that's such a great question. I love it. And, the term that we use is antagonistic pleiotropy, which basically, for, your listeners who may or may not know what that means, it means that there's a group of genes that do good things that, over time become a negative influence in general. And certainly the Wnt genes are some of those, as you mentioned. They're very important in development and in, formation of who we are as organisms, but then go awry in cancer. And so, what was fascinating for me is when I joined, the work actually started at the National Human Genome Research Institute under the scientific direction of Dr. Jeff Trent, who was the scientific director at the time, along with Francis Collins, who just retired as director of the NIH. But when I joined Jeff's lab, he said to me, oh, we need a cell biologist to work on this project in melanoma. And at the time I was doing prostate cancer, and I was like, oh, I really want to do, X in prostate cancer. And he said, okay, you can do that, but will you just help with this project? And so that's when I started working on WNTs and melanoma and became so fascinated by them for exactly all of the reasons you just mentioned, because they were such a double-edged sword, right? So, and, it's now 20 plus years later, and we're still finding new things about these genes and the way they work. So for example, very early on in my career, we identified WNT5A and we said, here's a gene that drives the metastasis of melanoma. It drives the way these cells move and how they get to the distal sites. And so in my head, I always sort of equated that. But now in our latest work that will hopefully be published from the lab very soon, completely unpublished data right now, we've seen that actually those cells that have that high expression of that gene WNT5A, they do need it to leave the primary site, but if they get to a distal site, they won't grow. So they get there and they need that gene to get there. They need that gene to survive there, but it also stops them from dividing and growing, which is fascinating. And then as we age, whatever is happening in the environment around those cells that have kind of implanted in that distal organ, let's say the lung, and they're sitting there in that distal organ basically asleep, there are changes that happen in the fibroblasts, the normal cells around them that now inactivate the WNT5A gene, and those cells grow out like crazy. And so this is really an underlying cause of what we call tumor dormancy, where a patient will get a tumor removed very early on in their history of the disease, and they'll think everything is fine, and then 25 years later they present with metastases in different organs. And so we think that this is one of the mechanisms that underlies that. So it's been absolutely fascinating.
Absolutely. And it's following that work, I mean, it's great to see you're still looking at that, but you leave National Institute of Aging, you head up here to Philly, down the street here at the Worcester Institute with my friend Dario Altieri, and you go to work, you're leading the tumor microenvironment metastasis program. And once again, here's something that this term microenvironment, it really seems like it's evolved a lot. We go from cells and we realize, okay, there's more than just cells around this tumor. They're secreting all sorts of interesting substances. There's an extracellular matrix there that does its own thing. There's blood flow coming in. And then on top of all that is an area that I'm personally fascinated with, that you also are a thought leader in, the biophysics of this tumor, shear stresses, membrane potentials, all these other things that are going on. They're not sort of biological component, but physically as you look at both sort of the tumors themselves and then the sites they go to, I said there's two different types of microenvironments there. How do you, in leading a tumor microenvironment lab, how do you decide sort of what the key parts in this microenvironment are in 2022? Because there's so many of them. Walk us through a little bit about, after you come in the office in the morning, what target do you decide to work on within the microenvironment itself? Because it just seems so multidimensional what we understand today.
It's so true. So, I think what's always driven us are the data, right? And so, and also just, I never think of myself as someone who's very smart, but just someone who's very practical. And so one of the first things that led us to even start looking at this was the fact that a melanoma cell spends its life at sort of this intersection between the epidermis and the dermis. And the dermis makes up this big chunk of the skin. And the cell type that is predominant in the dermis is the fibroblasts. Right? And what we know about skin fibroblasts, for example, is that as we age, they age with us. And they're not a cell type that's constantly turning over like the gut, for example, that, it sloughs off and then it's replaced. So it's a cell type that's actually getting old. And so my question when I first started that was, here's a cell type that's getting old. It's changing. If some of them die, there's no turnover, there's no replication to, some beautiful work out of Yale from Valentina Greco's lab showed us that those old that when a fibroblast dies, the neighboring fibroblasts kind of spread in and take over, right? And so practically for us, we said, okay, let's look at the fibroblasts. Let's look at what they're secreting. And so it's kind of a back and forth. So we decided on the cell type we were going to investigate. But then in order to say what are the targets that are interesting, we did a mass proteomic study, right?
Okay.
So like a generated a bunch of different targets. And then things that were interesting, different members of the lab said, oh, I want to look at that, or oh, I want to look at that. And, and patterns come out of these data. So one of the big patterns was the biophysical changes. It was very clear that in young and aged skin, the biophysical matrix was changing. And we know that if you look at an elderly person, they have paper thin skin, they have wrinkles. You look at a young person, they have nice, firm, wrinkle-free skin. So you know that there are physical changes. And again, that's the practicality, right? You can just look at skin and see there are these clear physical changes. And so the question we asked was, what are those physical changes? And what does that mean for a tumor cell that's embedded in one matrix versus another?
Excellent. And are there, because I think of melanoma, okay, I mean, one of the, terrible survival rate. I mean, you're working on the toughest of the toughest cancer there. But we can all get melanoma depending on, obviously, a bunch of different genetic factors, and obviously age impacts that. What are you learning? Because once again, I had Judy Campisi on a couple months ago, actively involved in sort of studying sort of the SASP in terms of the senescent cell, the secretory phenotypes, and this is another area that sort of evolved a bit. We used to think a couple of years ago we heard that SASP, all this stuff that these aging fibroblasts are secreting that are really bad, but now when we're looking at this sort of asking now, wow, there's a lot of stuff there. Are there specific classes of secreted compounds, certain ones that are of more interest than others? Because, at the same time that these secretory components that are negatively infecting, there's other possibly beneficial things there that help with wound healing and all the other good stuff. Talk a little about some of the stuff when you drill down into the aging, we'll call it the phenotype or the biochemical phenotype. What are you learning?
So we're learning multiple things. So the first thing we're learning is that the skin does not age in the same way the lung does. So the aged skin fibroblasts secrete completely different things than the aged lung fibroblasts, which look more like aged liver fibroblasts, but then lymph node fibroblasts look more like skin fibroblasts. So we're learning and that was kind of a bit of a shock to me to see this like complete organ specificity, but just not organ specificity, but the way they age is organ specific. So that has been absolutely fascinating and almost a dichotomous. So for example, we talked about Wnt5a, right? One of the other members of that family include the secreted frizzle related proteins. And we see in aging skin, SFRP2, which increases Wnt5a is secreted, but in the lung, SFRP1, which shuts off Wnt5a is secreted. And so it's this crazy balance of these different factors. Factors that seem to play a critical role that are secreted by young versus aged fibroblasts are the ECM factors. Those also change from organ to organ, but not as dramatically as some of the other things like the SFRPs, for example. And so what we're finding is that there is this consistent story of an aging phenotype that occurs that tends to be very organ specific. And our latest research is looking actually to see, is there a difference between male and female? Do they age differently? Because we know that the rates of melanoma are quite different in aging males versus aging females. And so, we wanted to drill back down. They also respond to therapy differently. And so we wanted to drill back down and ask, are there differences actually in the fibroblasts from different sexes? And that's been fascinating. So we find, for example, you mentioned Judy Campisi, who is my hero and who totally set me on this path in the first place. But we see that young female fibroblasts, for example, are more senescent right from the get-go than young male fibroblasts. But the rate of change, it's sort of that old in the 80s, there was that book, Men Are From Mars, Women Are From Venus, right? And then there was a lot of pushback against that book that said, well, that's, a little sexist and we're all the same. We just have some differences. And that's kind of what we're finding with the fibroblasts is that it's not necessarily the quality of the differences, but the sort of rate of change, if you will.
Mm-hmm.
So for example, both male and female fibroblasts will undergo senescence, but the male fibroblasts will sort of go from a much lower basal level to a much higher level of senescence than the female.
Fascinating. Really fascinating. As you study these microenvironments, obviously, one of the things I mentioned at the beginning, you're very focused on looking at sort of, here we are in this era of the smart targeted therapies, the immunotherapies, but, there's still segments that, they work great on here, no effect here. You're studying, what's going on in the microenvironment that's maybe causing these differences. What are some of the sort of the topics or the kinds of therapeutic interventions that you envision when we're specifically focusing about targeting microenvironments? Obviously, Judy talks about, her hot thing is senolytics and sort of getting rid of senescent cells that are secreting bad things. There's other things possibly we can do here in terms of epigenetically modifying cells. What is exciting to you that's coming down the path and saying, hey, these could be wonderfully synergistic with these smart therapies to make them work that much better?
So it's as if I emailed you that question before or something, because it's
I don't have it
question.
We'll talk later.
It's one of the things I'm most excited about right now. So last year we published a manuscript, and that manuscript showed, so in melanoma, there is a gene that is a driver oncogene, the BRAF oncogene.
Okay.
And we have amazing therapies that are targeted to that oncogene. So it's a mutant BRAF, and we have a drug that targets mutant BRAF, right? And so in a perfect world, nothing else should matter. Mutant gene, drug that shuts it down, done. However, if we take genetically identical tumor cells that have that BRAF mutation, and we put those cells half in a young mouse, half in an aged mouse, and we treat them with that drug, the outcomes are completely different. And the tumors in the aged mice, they act like the drug isn't even there. They just keep growing and growing and metastasizing, and it's like you never treated the mouse with a drug. And so we wanted to know why that was. And we did a profile of the secretome of the young and aged fibroblasts, again, to try to understand what they were secreting that could be protecting the cells in an aged microenvironment. And what we found was that they were secreting all these different types of lipids. And those lipids activated a fatty acid transporter in the melanoma cell. And that fatty acid transporter, when we shut it down in tumors in young mice, nothing happened. They continued to grow. When we treated them with the drug, they responded as they did normally, and then they came back. The same tumors in the aged mice that just grew through the therapy initially, when we first shut down that receptor, that fatty acid transporter, they went away and they didn't come back once we treated them with the drug. And so that is a molecule that was induced by the aged microenvironment that we could now target beautifully with adjuvant therapy against that FATP2 transporter and show that we could eradicate these tumors in older mice.
Very exciting.
So that's definitely, so it's, and then there are other targets. For example, we've shown that there are targets that drive angiogenesis, and we can target those in the microenvironment, shuts down angiogenesis in the tumors and, disrupts them. And those same targets that drive angiogenesis are, independent of the targets that are currently being targeted by anti-angiogenic therapy.
Got it.
So it gives us another reason why that's not working maybe in older patients.
Got it. And on the sort of the biophysical side of things, obviously a little more difficult to drug there, but I'm thinking, a couple months ago I had Mike Levin on from up at Tufts, and he's very hot on membrane potentials and things of that nature. Any interesting findings in your group in terms of how you may be able to deal with sort of these, I call them the sort of the levels that are sit above sort of the biochemistry in terms of some of these forces and stresses and so forth that also impact the microenvironment? Or is that not a focus of your group?
No, it is. So we actually have seen that there are molecules that are important. They, for example, they act as the knitting needles that knot together collagen and elastin in the skin.
Got it.
And those knitting needles get lost as we age. And so there's a question of how do we reconstitute them if we have, the molecule itself is called haplin-1. And if we have haplin-1 in the skin, we see that, you know, the skin is young and firm, you know, so a lot of people, whenever I give that talk, ask me if they can have some haplin-1 for their wrinkles. And I say, no, you have to wait till I can do it for myself first. You know, but when we do that, we also inhibit the ability of tumor cells to move from that primary site to the metastatic site, because that biophysical matrix kind of comes and locks those tumor cells in. But also what we've seen is that, you know, with the changes in the biophysical matrix, they affect things like the integrity of blood vessels. They affect the ability of immune cells to come into the tumor microenvironment. And by manipulating, you know, different age-related aspects of that immune microenvironment, our hope is that we can both deliver more effective therapies through these blood vessels, as well as inhibit metastasis.
Excellent. And actually, I know that you have some very staunch, very serious feelings towards tanning, and you know, you had your program here in Philly with the sun lotion. Any other public programs, public educational things you're doing on that front still? Because I know that's near and dear to you as well.
Yeah, so there are a lot of things. You know, many things were sort of stymied by. I apologize for the ambulances in the background. There are many things that were stymied by the pandemic, of course, a lot of the community outreach. You know, we are interested in working for, so there was a campaign with the Melanoma Research Foundation where they taught hairdressers how to recognize moles on the scalp, for example, right? And so it turns out that people of color get melanoma in their nail beds and the soles of their feet and the palms of their hand. And these tend to get ignored. In fact, that's what Bob Marley died of. He died of melanoma in his toe. And, you know, so a few years ago, I was getting a pedicure and I looked down and I saw on my toe a classical, you know, it looks like someone took a Sharpie and drew a straight line on your toenail. And I was like, oh crap, I know what that is. And it's not good. And I went straight to the amazing dermatology group at Penn, and they removed it. And I was very lucky because it was completely benign. You know, it occurred to me like, well, you know, just the way they train hairdressers to recognize like moles on your scalp, why can't we train manicurists and pedicurists, especially in salons with people of color? This could be a really important, you know, they could notice this and say, hey, you should go see your dermatologist. So, you know, so that's a campaign that right before the pandemic, I was gearing up to start, that we never really got off the ground. And now that, you know, well, I don't want to say now that things are calming down, because they don't, they don't seem to ever calm down. You know, we would like to do that again. And also now here in Baltimore, it would be nice to see if we could do the sunblock dispenser program here as well. So, and that was something really spearheaded by Mike Schmidt and the RDK Foundation over there in Philly. So.
Got it. Well, hopefully we'll see more of that as we come out of this mess. But just a couple other questions for you. I know you have a lot going on. You know, we were talking about Judy Camp PC before. You know, one thing that you both have in common, she's your hero, but at the same time you have a similar hero, Mina Bissell, who you both respect quite a bit. Mina Bissell, when you read sort of her early materials, her hero, we just lost a couple of weeks ago from here in Philly, Beatrice Mintz, who just passed away at the age of 100. I'm just going to loop back for a moment, because we were at embryogenesis, we came through aging, and I just want to go back to one thing and get your thoughts on it. Obviously, Beatrice Mintz did the seminal work here in the Philly area back in the 1970s with, you know, creating embryos from tumor, you know, the teratocarcinoma cells. And we have this, you know, interesting paradox that, you know, we don't think of all the gene mutations and so forth that happened in those 9 months where we're inside our mother's womb, but there's a lot of cell division, a lot of mistakes going on. Any interesting, because I know that sort of seminal work in saying, wow, there's some very interesting stuff that happens in the embryo that, you know, normalizes cells that were very bad to begin with. Any interesting work on sort of the embryology front, whether it's your group or groups that you partner with, in sort of further understanding what are some of these interesting normalization forces in an embryo that don't exist much later on in life that maybe we could go back to and take a second look at, because I know that was 50 years ago, but nonetheless, it's pretty groundbreaking work still.
Yeah, I mean, I think there's so much interesting work that's being done there. You know, it's not something we do at all, but certainly the Drosophila communities, the C. elegans communities, you know, some of the amazing model organisms they have to study embryology have just been incredible, right? A lot of the Nobel Prize winning discoveries, you know, have come out of some of these embryological studies. And certainly what we're learning from embryology applies to cancer, because many of the genes important for division and growth and halting of division and growth become defunct in cancer, right? Yep. And so understanding embryology is critical to understanding cancer progression.
It's always been a sort of fascinating area just to watch that over the years, and embryo is a very unique microenvironment in its own right, and so
yeah,
but it's interesting. Actually, one final question for you, I got to ask, but in 2021 September, you're appointed to the one of 7 researchers, National Cancer Advisory Board. What was that? Did Joe Biden just call you up and say, Ashani, I want you on the board by the 5th? I don't know, how did that work? What exactly went on?
It's actually kind of a funny story. So I got an email from the White House, and I saw the email from the White House, and I was like, these scammers will try anything.
Exactly.
I mean, this is clearly a hoax, because who's going to ask me to be on their National Cancer Advisory Board? No, it didn't even say that. It said, because if it had said something about the NCAB, I might have taken it seriously, but it said you are being considered for a presidential appointment to the White House by President Biden. And I was like, oh, okay, so I suppose you want my social security number next, you know?
Send $500.
And I just ignored the email, because I was like, clearly this is a scam. And then later I was like talking to my husband, and I was telling him about it, and he's like, let me see that. So I was like, okay. And then he looked at the address, and he's like, Ash, this is a .gov email. You might want to respond. So, yeah, I mean, I have to tell you that when it hit me that it was real, I was so overwhelmed with this incredible flood of emotions, right? Because I'm someone who came here expecting very little. I moved here from Africa, and now the President of the United States is asking me to be on his advisory board. It was mind-numbing to me. And I have to admit, like, when it sank in, I sat down and I cried for half an hour.
There you go.
And then I called my mom. Like, I wasn't allowed to tell anyone, but when I was finally allowed to tell anyone, the first person I called was my mom. And she, like, further, like, made it worse, because she said, I'm so proud of you. And the thing I'm most proud of is that you've always remained such a kind and nice person through it all. And I was like, oh my God, that's it. See, yeah. I'm tearing up again. So,
no,
it was a huge shock. And I cannot describe like the level of an honor it is for an immigrant like me to come all this way and, have a voice here.
It's a beautiful story. It's fascinating. Really, I'm rooting you on everything that you're going to be continuing to do there on all these fronts, and really wishing you the best with all of it. For everybody that is going to be listening to this episode across our different podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Ashani Weeraratna, Bloomberg Distinguished Professor, Chair Biochemistry and Molecular Biology, Professor, Johns Hopkins Bloomberg School of Public Health. Pick up a copy of her book, Is Cancer Inevitable? She's also, of course, on the National Cancer Advisory Board, recently appointed by President Joe Biden. Ashani, I want to thank you for taking the time out of your schedule to come talk with us about all these topics. Obviously, thank you for everything that you do and continue to do there. And as we say on this show, thanks for helping to create a better tomorrow through what you're doing. It's really fascinating. Really great.
Thank you so much. And thank you so much for having me.